Superhumans could be modified with a far better tolerance to lower temperatures in the future. According to new research, experts found the key to resisting the cold is hidden in the brains of all mammals, and they believe it could be activated in human cells. The finding would also let people enter a hibernation state similar to some animals, which could be useful on long journeys to deep space. Researchers from Yale University found that hibernating animals hold the key to why some species are more sensitive to the cold than others. Hibernating rodents have evolved cold-sensing neurons with diminished ability to detect temperatures below 20 degrees Celsius. The TRMP8 gene is the specialized area in the brain responsible for reacting to the cold when it is triggered. An adaptation to this strand of DNA lets hibernating rodents' body temperature drop for long periods of time without causing them to feel stressed by these conditions, triggering their seasonal slumber. Animals like ground squirrels don't feel the cold even when it's snowing outside, and another rodent, the Syrian hamster, shares the same mechanism. Researchers discovered that this ability could be manipulated in other animals, including humans. Speaking to study co-authors Dr. Svyatoslav Bagrintsov and Dr. Elena Grakova said, future research will make it possible to confer cold tolerance to human cells, with potential implications for medicine. Understanding the hibernation phenomenon in general will pave the way to inducing hibernation in humans, which seems necessary for long-term spaceflight. While humans and spaceflight is the long-term goal, the next step involves genetically altering rodents. The authors continued, at this moment we are working on generating a squirrelized mouse. We are trying to make a non-hibernating species of mouse cold-tolerant by substituting the mouse TRMP8 gene with the squirrel gene. We expect that such genetically modified mouse will develop tolerance to cold. The temperature at which TRMP8 gets triggered varies massively from species to species. This study found all rodents have receptors on brain cells known as somatosensory neurons that sense cold, but the chemicals in ground squirrels and Syrian hamster take a lot more to be activated compared to those in animals that do not hibernate. In order to compare the biology of the different types of rodents, the researchers performed tests on ground squirrels, hamsters and mice. Scientists put the rodents on two temperature-controlled plates, one warm at 30 degrees Celsius and another cooler one, which they varied from 20 degrees Celsius down to 0 degrees Celsius. The animals had the choice to move between the two plates, and the mice strongly preferred the warmer plate. Mice always strongly preferred the warm plate, but the temperature change made no difference to the ground squirrels and hamsters until it reached a cool 5 degrees Celsius. Dr. Grakova's team connected this behavior to a chemical pathway in neurons called TRPM8, which, when triggered, leads to the sensation of feeling cold. TRPM8 is a protein on the outside of specialist sensory cells in the brain which control the flow of charged sodium and calcium ions in and out. This flow causes a charge to build up, which then fires off an electrical impulse, continuing the signal. In ground squirrels and hamsters, TRPM8 is less sensitive to cold than the same chemical or ion channel in mice. Dr. Grakova added, this process is very complex, and TRPM8 is just one part of the mechanism.